Welcome and thank you for standing by. I would like to remind all participants the call is being recorded. If you have any objections, you may disconnect at this time. I now want to turn the call over to Ms. Diane St. Germain. You may begin.
Thank you. Hello, I'm Diane St. Germain with the National Cancer Institute. On behalf of the International Society for Quality of Life Research and the National Cancer Institute, I would like to welcome you to the webinar series titled Best Practices for Integrated Patient-Reported Outcomes in Oncology Trials. The webinar series is comprised of 6 webinars, each approximately 45 minutes in length. The webinars are designed to be viewed independently to meet your individual learning needs, or the series can be viewed sequentially in its entirety. Today's webinar is the first in the series and is titled How to Identify the Patient-Reported Outcome Context for Clinical Trials and the Relevant Patient-Reported Outcome Domains, presented by Dr. David Sella. I will now turn it over to Dr. Sella.
Thank you. This is Dave Sella. I am professor and chair of the Department of Medical Social Sciences at Northwestern University's Feinberg School of Medicine. I'm also associate director for cancer prevention and control at the Robert H. Lurie Comprehensive Cancer Center at Northwestern University. And as Dr. St. Germain mentioned, my title is How to Identify the PRO Context for Clinical Trials and Identify the Relevant PRO Domains. It's really the launching webinar in the series of six webinars to sort of set the table. So the first thing I'd like to do is distinguish and in that way identify and define patient-reported outcomes, or PROs, from other assessments that are used in clinical trials. A patient-reported outcome usually refers to any standardized assessment that's based on the patient's self-report, and that is typically or ideally completed alone by the patient without interruption. And some examples used frequently in oncology include the Medical Outcome Study SF36, the PROMISE panel of measures, the EORTC QLQC30, the FACT and FACET family of measures, and the PRO CTC-AE. In contrast, to the right are clinician-reported outcomes, and these are assessments that are based on physician or other provider interpretation of the patient's status. Typically used in oncology are the Eastern Cooperative Oncology Group Performance Status Rating, or ECOG-PSR, or the Karnofsky Performance Status Rating. In addition to these ratings of health done by patients and providers and PROs and ClinROs, there are performance-based assessments that are sometimes used, and these are objective tests that measure patient performance, such as a stair climb or a 6-minute walk test or FEV1 lung function test, or objective performance tests from the NIH toolbox, such as motor, sensory, and cognition tests available from that panel of tools. And then finally, there are standard, more typical, if you will, clinical assessments used in oncology that are often based on clinical examination and diagnostic evaluation, such as the patient's disease stage, histology, pathology, radiography, et cetera, in response to disease, progression of disease, and the like. When we compare performance measures, such as in this case a bicycle treadmill type test versus a self-report, patient reporting on his or her own physical functioning, these are conceptually related concepts, but they tend to agree only around a 0.4 correlation. That is, they share some variance. People do a good job of reporting how they're performing physically, but what they say about how they're doing does not always agree completely with actual tested performance, and there are a lot of reasons for that. Rather than say one is more valid than the other, because we really don't know which one is more valid, we consider both to be providing important and correlated, but also somewhat distinct information. So they both have value in the clinical research setting. Now, to get started in our webinar, in this first webinar, I'm going to talk about 2 related considerations through the course of this presentation. One is the PRO context, that is the surrounding information and aspects that are important to keep in mind when you're evaluating patient-reported outcomes in clinical trials, and then how to select, identify the relevant patient-reported outcome domains in a clinical trial. When it comes to identifying the PRO context, there are some basic points that we should keep in mind. First is that the context for a truly patient-centered PRO is, by definition, centered around the patient. That is to say, we really want to make sure we always keep in mind that if we want to understand the patient's perspective, there's really no way of getting around the fact that we have to ask the patient on his or her opinion. The second thing is that a fully individualized patient experience of treatment is really not realistic or easily aggregated. We recognize that all people come to cancer and cancer treatment from unique perspectives, and it's very difficult to summarize everyone's experience with one standard set of questions, but by the same token, there's such variety in the individual experiences of people that trying to over-individualize is really met with very limited success when people have tried to do this. A third thing to keep in mind is that global evaluations, that is questions like overall, has the treatment been effective, or overall, how's your quality of life, they're useful pieces of information, but they tend to be insensitive to change, and they're often biased by emotional factors and other what we might call distracting influences in the context of identifying the value of a cancer treatment as being more or less effective than a comparator. Finally, virtually every treatment setting in oncology has trade-offs of benefit and harm. It's difficult to find any example of a treatment that has no risk associated with it. We need to keep these context factors in mind as we consider studying PROs in a clinical trial. This is a patient who wants her dose adjusted because she's not nearly as happy as the people in the ads. That is to say, what we might know in the general overall group conclusion about a drug effectiveness or treatment effectiveness does not necessarily apply to an individual patient. Here's a person who's completed an endoscopy and is very happy about his results with the patient, saying everything's wonderful, but little does he know unless he asks that there are plenty of things on this patient's mind. And these things on the patient's mind about the bother of symptoms and trouble sleeping and worry and fear and just being generally unhappy with his condition are only going to be found out if we ask the patient. So all of this is to say we really must ask the patient. That's the first point that we talked about keeping in mind. And the steps toward doing that, the first step is to get the right questions. That's easier said than done due to points 2, 3, and 4 that I mentioned about how difficult it is to truly individualize, etc. And the questions, therefore, should be a sampling of the most important symptoms and concerns of patients that are in the same boat as the person or the trial that's being considered. And this, in essence, is the context that we're talking about, that is being in the same boat. We want to understand what are the most important symptoms and concerns of people with a similar condition, a similar type of cancer, similar context or setting of treatment. I'll come to these points in more detail in a few moments. The second step is to come up with some way to summarize or score these individual responses that we've decided are the most important concerns to be assessing, and report those results in the larger context and to place them into the larger, broader context of outcome in the trial. That is to say, we don't look at PROs in isolation. We really need to look at them in the larger picture. So, moving now to a specific understanding of what PROs are. As I mentioned, they are reports that come from patients. The vast majority of PROs that are used in clinical trials fall under the heading of health-related quality of life. And this concept of health-related quality of life includes disease symptoms, treatment side effects, the functional status of the patient, that is what he or she can do physically or mentally or socially, and the patient's overall well-being, mood, or general health perceptions. However, health-related quality of life and its domains are not the only patient-reported outcomes that we're interested in and sometimes collect. We also collect things like treatment satisfaction, treatment convenience, medication logs and diaries, the medical resource utilization information that patients can provide about need for ancillary care during the time they're receiving treatment, and the cost of therapy, both direct medical costs and indirect costs that are borne by the patient to be able to go through the treatment. Health-related quality of life, the largest segment of patient-reported outcomes, refers to the extent to which one's well-being is affected by a medical condition and or its treatment. So it really incorporates both the treatment and the condition that's being treated in what it's intended to cover. It's a multidimensional concept that covers physical, social, and mental areas of life, and it's also a subjective concept, meaning that, as I've been saying, we really want to capture this information directly from the patients and not have it filtered through family members or other healthcare providers. One of the best conceptual models, and one that's stood the test of time now, having been available for nearly 20 years, of health-related quality of life was published by Ira Wilson and Paul Cleary in the Journal of the American Medical Association. And it is a sort of left-to-right moving diagram that starts on the left with some sort of biological and physiological set of variables. In this case, it could be a certain cancer at a certain severity and perhaps a treatment being received for that cancer that has physiological effects. Those are considered outside the realm of health-related quality of life. But then as we move to the right from that sort of biological and physical starting point into these domains of symptom status and functional status and general health perceptions and global quality of life and satisfaction, this is where we're talking about this concept of health-related quality of life. It encompasses all of these concerns and all of these issues. The response or reports that we get from patients about these different things, about symptoms and function and health perceptions and global evaluations of life quality, are influenced not just by the biology of the disease or the treatment, but also by characteristics of the individual himself or herself, their own sense of optimism, their negativity or positivity, their willingness to report problems versus a stoicism. All of these individual personality factors influence the way people respond to questionnaires, and also characteristics of the environment, such as socioeconomic status or neighborhood conditions, or things that can influence the way people feel their lives are hindered or enabled, that are relatively independent of any particular disease or treatment they might be receiving, but might affect the way they respond to some of the questions that we ask. So this is a fairly straightforward, linear way of viewing health-related quality of life that can help in the beginning stages of putting together a sense of an endpoint model in a given clinical trial that you're planning. So the key steps when you're developing a successful measurement strategy are first to understand the disease itself and the expected treatment effects. That is, what's the environment, the treatment environment that you're dealing with. The second is to identify outcome domains that are relevant to patients and to this disease area, which symptoms are important, which functional abilities are important, et cetera. A third is to articulate an endpoint model and a conceptual framework, and I'll say more about what these are in just a moment. A fourth is to select or develop PRO instruments to measure the domains that you've determined through literature review and study of the patients themselves are important. And finally, fifth, to implement the PRO study in a well-designed clinical trial so that it's integrated into the trial. A patient-centered PRO context would ask the following questions. How are treatment options presented to and understood by the patient? So we know in oncology and various types of cancer, lung cancer, breast cancer, et cetera, there are certain options that are available and hopefully discussed with patients. And how is it that they're discussed and how are they understood by the patient? What are the goals of treatment? Do the provider goals match the patient goals? We hope so. but it's worth asking and finding out and determining the extent to which that's the case, because if they don't, the patient's outcomes are going to be more driven by the patient's expectations and goals than they are going to be by the provider's goals. And finally, how does one evaluate whether treatment goals are met or which therapy is better? Do we look for the therapy that's more effective on a clinical parameter, such as tumor response or progression-free survival? Or do we look for a therapy that's less toxic, with an assumption, perhaps proven, at least demonstrated in the trial, that the clinical endpoints are similar or not significantly different from one another? Then we might pick the treatment that has less or fewer side effects. Do we look for a match or for an agreement of the clinical and the PRO outcomes? That is, if the clinical outcomes are better on one treatment, do we also want or expect the PRO outcomes to be better or at least not worse? Because if the clinical outcomes are better and the PRO outcomes are worse, it becomes a very difficult calculus to put together and to balance and make a final risk-benefit or benefit-harm appraisal. Do we add up scores and compare them to one another across arms? Which scores do we add up, and what weights do we apply to those questions that are being answered? Some questions may be more important to patients than others. Some questions may be more important to some patients than they are to other patients. And we might wish that we could, as I said at the beginning, individualize scores by patient preferences, but it's a very difficult thing to do, and so far has not yet been mastered in this area. So we tend to apply constant weights and sometimes simply equal weights to the items or questions that are being answered. We do know that a global evaluation won't suffice. Remember, that was point 3 and the points to recall or keep in mind when considering the context of a PROs in a clinical trial. Global evaluations are nice, but they are usually insensitive to change, and they don't always tell us much about the specific and more direct impact of treatments on the patient. So I mentioned this term endpoint model. What is an endpoint model? Well, endpoint models will articulate the associations or relationships among different health outcomes. They are, in essence, links between the clinical outcomes and the patient-based outcomes. An endpoint model, by linking and associating different outcomes to one another all on one page, will demonstrate, ideally, the importance and association of these different health outcomes. It's not technically a theoretical framework or theoretical model, but it does begin to move toward that direction by saying these are the associations and linkages that are probably going to be considered important in this particular treatment setting. Usually the model is driven by the patient population, that is what disease primary site, stage, histology, etc., and the disease characteristics, including the symptoms and functional impact of the disease, as well as the expected treatment effects. So you put all these different things into one two-dimensional model to try to express visually what you're interested in comparing and looking at across treatment arms. The endpoint model provides the rationale and framework, if you will, for the patient-reported outcome measurement strategy, which is accomplished by identifying the important PRO domains and then explicating the association of these domains with treatment benefit. That is, if a treatment's effective, it should affect X, Y, or Z, and maybe not A, B, and C. Usually, we think in terms of proximal versus distal outcomes. That is, that we expect treatments to have a stronger effect on outcomes that are more proximal to the treatment we're talking about, and a weaker effect on those that are more distal. A very obvious example would be a pain intervention, an analgesic given for pain. We would expect a benefit to pain. It might also help the patient's fatigue that's associated with pain and may even help their mood because of relief of pain. But the impact on things like fatigue and mood would not be as strong in a predicted endpoint model as the impact of treatment on the pain itself. The endpoint model also informs the identification of the PRO targets and hypotheses. That is, you typically find targets that are more proximal to the intervention being delivered and that are more directly logically related in a hypothesis to the effect of therapy. So let's show you an example, and you might notice that this figure, in a sense, mirrors the chain from left to right of that Wilson and Cleary model that I showed earlier. But here we have advanced ovarian cancer. That's the biological and physiological side of things. And let's also imagine that in that advanced ovarian cancer box on the left, we're also talking about some kind of treatment, say intraperitoneal versus intravenous chemotherapy for advanced ovarian cancer. Now, we might expect very proximal to that intervention, either one of those interventions for ovarian cancer, that there would be some experience of abdominal pain and discomfort and ascites that could be caused by the disease and ideally relieved by treatment. Those, in turn, would logically have an effect on things like appetite, early satiety, fatigue, and sleep problems, which over time would logically accumulate into trouble getting around with basic mobility, activities of daily living, and general physical function. And they might even have an effect on leisure activities and social function and getting along with family and friends and in being able to enjoy one's life and one's mood, one's emotional well-being. All of these things contribute to a global impression of quality of life. The more proximal abdominal pain and discomfort and ascites are the outcomes that one would expect more likely to be affected directly by the chemotherapy treatment. And the more distal outcome of quality of life over on the far right might be affected by differences in treatment, but the probability seems lower in most cases. And this kind of a model can help one to form hypotheses and then select endpoints on the PRO panel to assess. So in order to get the right questions, sometimes people feel they have to construct an instrument. It's better, obviously, if you can find one off the shelf and use an already validated instrument, and very often that's the case. But when one has to construct one, this is a typical set of steps one goes through to do that. One, first, to establish the target population, the scope of the assessment, and the concepts to include, as well as available resources to do so. So, for example, with the ovarian cancer example provided earlier, you might pick those advanced ovarian cancer patients and those different items, those different domains in the endpoint model as the range or scope of the things that you want to assess in women with ovarian cancer. Then you develop items based on a literature review, looking through the literature of others who have studied this and what they've identified as key issues. Perhaps conduct your own focus groups and or in-depth interviews, and there gather the very important information directly from patients about what's important to them and what they notice and would want to see assessed, and that should ideally line up with the literature review, although might point out new areas of content that weren't covered by prior literature. And then take that set of questions or domains and pretest in a sample of that target population, in this case advanced ovarian cancer, and go through a series of qualitative and quantitative steps to reduce the items to a manageable number, to evaluate the psychometric properties, and then to evaluate even their cross-cultural equivalence, because in many cases you'll want to be studying women from different cultures in one multi-center trial and sometimes even multinational trial. And then after that, you want to pilot test this candidate instrument, which can also contribute back in a feedback loop to further item reduction or modification, and then finalize the instrument through this process. Then you're ready to have something to test in a clinical trial. Now, a measurement model, which can be articulated right from the endpoint model to give you a framework for measuring important patient-reported outcome domains. And just sticking with ovarian cancer here as an example, we have items or questions that might ask about appetite, nausea, vomiting, bloating, shortness of breath, abdominal fullness. And these items should, in a setting of ovarian cancer and advanced disease where ascites is often a concern, these should correlate with one another sufficiently to be able to form something of a scale or a set of questions that cluster together well and correlate with one another sufficiently to sum up into one number. So you don't have 6 scores, you have 1 score that you could call a score for the domain of ascites. You can take another 3 questions on heartburn, feeling discomfort in the stomach area and discomfort in pelvic area, using words that came from those patient interviews and focus groups, and summarize them instead of having 3 scores, having 1 score that could be called abdominal pain and discomfort. Recall that these are the 2 domains that were most proximal to ovarian cancer and treatment in that Wilson and Cleary model. And you could operationalize those 2 endpoints as your primary PRO endpoints and have these 9 questions summed up into 2 scores, and that would be those would be your endpoints to test any group differences in the treatment comparison. So this process of instrument development and evaluation tends to take place outside of the trial in the beginning and then migrate into the clinical trials in terms of to determine their real-world relevance and utility. Instruments that are based on the sampling of a universe of items covering a domain are usually the way to go in terms of instrument development, and we want items to represent as many, if not all, aspects of that domain, and also that we want the instrument development work to have a content map that guides the construction of items to measure that domain. Now, in terms of psychometric evaluation of instruments, we want to be able to test an instrument, a set of questions summed into a score to generate hypotheses about the structure of those instruments. That is, in those examples I showed, that those 6 items should correlate with one another sufficiently to be summed into a score, and also that they predict differences in groups that you would expect them to predict based upon the content that's being discussed, whether it's ascites or abdominal discomfort or something else. And when you go through this exercise of showing or demonstrating that these aggregated scores differentiate people that you would expect them to differentiate or are responsive to change over time, this is the process of psychometric evaluation and validation. So I'll talk a little bit about the conceptual model now around this building of an instrument, developing and evaluating it. You first want to be able to demonstrate in a convincing way that something you think you're measuring exists as a concept, and therefore you want to be able to link that concept with what you're asking about to measure that concept. And if you can show that the concept correlates and is linked with these different questions or measures in the predicted way, then you've created a convincing argument that you're measuring something that's worth knowing and measuring in this clinical trial context. There'll be much more said about this in the next webinar. To evaluate their instrument, there should be a correspondence between the items, that is the questions, the domain score, the rolling up of those questions into a summary score, and then the overall score. The expectations that you have should be stated in advance, that is prior to the validation process, so that you're in essence confirming a validation hypothesis when you're developing this questionnaire. The other thing you want to be concerned about is that you have asked enough of the right aspects or components of a given concept that you're evaluating. And we refer to that as content validity. And it's a process of being sure that we have done enough qualitative research and concept elicitation, whether through focus groups or individual interviews, to have been able to assure ourselves that we have gathered sufficient evidence for covering the full breadth of a concept, whether that's pain, fatigue, nausea, et cetera. And the concept elicitation part of content validity involves the generating of words and phrases from people, whether in focus groups or interviews. The instrument evaluation part of it is having developed the questions, frame them, to then make sure that people read and understand and comprehend and then answer those questions with the same interpretation and meaning as intended by the developer. And these two things are looked at together to make sure that we're, in essence, asking the right questions. This process of developing questionnaires uses the concept of saturation to ensure that we have talked to enough people and gathered enough information about what's important in order to be confident that we've sort of covered the map. People have defined saturation as data adequacy, have operationalized it as collecting data until no new information is obtained. And Margaret Mead actually referred to it as the boredom that occurs when investigators have heard it all. So you're not hearing anything new. There are specific methods for determining that no new information is coming through, so you may as well stop asking more people because you won't get new content, or the probability of getting new content is so low it's not worth the added effort. Why is it important? Well, scientifically, saturation is important because it helps establish the conceptual validity or the trustworthiness or truthfulness of the phenomenon under observation, that is, it really exists as we think it exists. It's practically important because it helps determine how many interviews or focus groups we need to conduct before stopping. There is no formal power analysis that one does with qualitative research. This is the method for determining sample size requirements in this kind of qualitative research. That's when you reach this point of saturation. There are some rules of thumb that range anywhere from 5 to 60 interviews required for saturation. None of these sources that has this pretty wide range of saturation, except the guest source, the last one, provided any evidence for their recommendations. Generally in oncology, the number needed for saturation falls somewhere in the range of 10 to 30 people, depending upon what's being inquired about. And what about focus groups? As opposed to using content solicitation by individual interviews, what about gathering content through focus groups? The rule of thumb there is something like 3 to 4 focus groups per any defined group is usually enough to have achieved saturation. Now I want to talk about evolving directions in assessment that get at this specificity of assessment, trying to find the proximal areas of the domains of interest and have more targeted hypothesis-driven clinical trial research than we've seen typically in the past. Now I'm going to talk about symptom indexing because symptoms, again, if you remember from the Wilson and Cleary diagram, are furthest to the left, most proximal to the disease and to the treatment that's being received in oncology. Now, some quality of life instruments, for example, the EORTC instrument and the FACT instrument, include cancer symptom items in addition to producing a total quality of life score or some sort of global score. And global quality of life scores, as I mentioned, may obscure important and significant changes in disease-related symptoms. So there's a bit of a problem with these instruments that they don't ideally compile or aggregate scores for these endpoint models as I've been talking about. And I'll come back to that. Now, today's best option for coming up with the right set of questions, what are the domains to choose in a trial to make sure that they're context specific, forces us to define upfront the breadth of the context that we're talking about. So, for example, do we want our questionnaire or our scale to be relevant to all people? That is, we ask questions that are important to everyone. Pain is important to everyone. Depression is important to everyone, et cetera. Do we want the domain, or do we want the sample or the population of interest to be all cancers? Do we want it to be even more narrow than that, just the primary site, just lung cancer or just breast cancer or ovarian cancer? Or do we want it to be primary site and stage, that is say, advanced lung cancer or early stage breast cancer? Or do we want the context of use that we frame our items around to be primary site histology and stage? We can get even more specific to primary site histology, stage, and line of treatment for second, third, fourth line. We can get even more specific and say we want this questionnaire or these sets of items to be specifically appropriate for the context of use of one primary site, histology, stage, treatment line, and genotype. And even then go further and say primary state, histology, stage, treatment line, genotype, and trial eligibility criteria. I think this last example is, some might say it's absurdly specific, but it is actually what sometimes we find some people in the pharmaceutical industry are compelled to go to because of a concern that it might be slightly different. The issues might be slightly different for people who are not eligible for a given treatment trial. And my personal preference lies somewhere in the middle of this continuum, that we're probably somewhere around primary site and stage, that's perhaps the optimal degree of specificity that we want to get in terms of the population of interest for a given set of symptom indexes. That is one primary site and one general stage of disease. So, for example, advanced ovarian cancer is a reasonable group and specific enough group for a context of use. And when we index symptoms, we're talking about nesting tumor-specific symptoms within multidimensional questionnaires, and that's what we have with instruments like the EORTC, in fact. And this creates the opportunity to derive targeted symptom scales that can be disease-specific, precise, and clinically very appropriate. Fortunately, we have now a guideline that's been proposed and published now in the Journal of the National Cancer Institute by Reeve et al. Just this year, recommending 12 core symptoms for adult cancer treatment trials: fatigue, insomnia, pain, nausea, anorexia, depression, anxiety, cognitive problems, dyspnea, neuropathy, diarrhea, and constipation. This doesn't mean that all 12 symptoms have to be assessed in all studies, but it does mean that here is a list of the 12 most common symptoms to consider in your clinical trials in adult cancer treatment trials, and make the case for which ones of this list are most important or more important and why, and make the case for which ones might be less important in a given clinical trial context, again referencing back to your endpoint model. Recently, we had completed a set of studies that included over 500 patients and 500 clinicians and produced 11 brief symptom indexes across these 11 tumor types: breast, colorectal, lung, prostate, bladder, kidney, ovarian, lymphoma, head and neck, brain, and hepatobiliary. And I'm going to just talk a little bit about the ovarian cancer one because that's the example we've been using, and not go into the details of the methodology, but just to say that we have sorted out these the top 9 in the green box, disease-related symptoms, one disease-related symptom that's emotional, that's in the yellow box, 5 treatment side effects, and 3 questions about general function and well-being. So you have components of an endpoint model: physical disease-related symptoms that are an aggregation of 9 symptom items, emotional disease-related symptoms, that's worry about condition getting worse, that's 1 question, 5 questions on treatment side effects, and 3 questions on general function and well-being. 4 components of a potential endpoint model in advanced ovarian cancer. And after having done all that, now that we think we have the right questions, how do we aggregate them? How do we score them? And how do we compare the results that we get on PROs to the other clinical data? How do we report them so they reflect their place in the full clinical outcome story? I'm just going to show you some examples. This is an example published by Chris et al. In JAMA about 10 years ago that showed that a lung cancer symptom score change was linked with tumor response in advanced lung cancer. The red line are patients whose best response was stable disease. The yellow line, patients whose best response was partial response, rather, so they're partial responding patients. And the green line are the patients whose best response was progressive disease. And Chris et al. Showed that even by 4 weeks, well before the radiography data had come in to show any response data, and even by 6 weeks, those patients who were partial responders had significantly better symptom scores than the patients whose best response was stable disease or progressive disease. So there's a harmony or a synchrony agreement between the clinical outcomes in terms of tumor response and the PRO outcomes, in this case lung cancer symptoms. Here's a study that also looking at lung cancer symptoms showed that the patients that had improvement in lung cancer symptoms survived much longer than patients who did not have improvement in lung cancer symptoms. So it's actually using the predictive ability of the symptom response data to, in essence, validate and confirm the value of symptom response. On this slide, this shows only the patients with stable disease. Here, the dotted survival curve in the middle, that's all patients in this trial with stable disease. The other two plots, the solid line, the better survival group, and the dashed line, the poorer survival group, those are subsets of the stable disease patients, this group in the middle. And the subset, that's the solid line, are those patients with stable disease who also had improvement in their lung cancer symptom score. And the bottom plot, the dashed line, are patients who had worsening or no improvement in their lung cancer symptom score. So all stable disease, but those who had improvements in their lung cancer symptoms lived much longer, even though their disease status was no better than maintaining stable disease. Again, showing value to the PRO. Now, here's a study that is related to progression-free survival, not overall survival, comparing axitinib and sorafenib in kidney cancer. And the point here is that there were no differences between axitinib and sorafenib over time in their kidney cancer symptom scores until the point at the end of treatment. When treatment was over, if you look at this group here at the end of treatment on the far right, the symptom scores dropped or got worse by a magnitude of about 3 or 4 units on average. It didn't matter which treatment they were on. There was no treatment arm difference here. But what it does show is that the end of treatment is a time of worse symptomatology on the part of patients, which helps to demonstrate the value of progression-free survival as an endpoint in clinical trials. Finally, here's an example from the EORTC instrument where 3 symptoms, cough, dyspnea, and pain, were compared in the erlotinib versus placebo registration trial for erlotinib or Tarceva. And the time to worsening of cough, dyspnea, and pain was longer, or that is better, on erlotinib versus placebo by a factor of 1 to 2 months. So the patients on erlotinib had 1 to 2 months longer period before those symptoms got worse than the patients who were on placebo. All significant and all supporting, again, the clinical endpoint of improved survival in this case. So to close, I want to offer some reflections on patient-reported outcomes today and tomorrow as you move into the rest of the webinar. First is that PROs, or patient-reported outcomes, reflect any content area that requires direct assessment from the patient perspective. A PRO, or a PROM as some call it, patient-reported outcome measure, used to be called health-related quality of life measures, and before that we called them quality of life measures, and before that we called them health status measures. Now today we think of PRO measures as more than just a set of health-related quality of life measures that are in the armamentarium, but that is the largest by far group of instruments in the PRO family. But we've used different terminology over the years, and they're all more or less meaning the same thing. The concept of health-related quality of life includes symptoms, side effects, functional status, well-being, treatment satisfaction, treatment preferences, and health health perceptions. So content of PRO certainly can be pretty broad and far-reaching, so it's important to zero in and narrow down what you're really focusing on in your given trial. Another point is that measurement without meaning is really worthless. We can measure lots of things, but we have to make sure that we're measuring something that has some validity, something that makes sense, that behaves in ways that we might predict, that we can analyze and interpret in the context of the larger trial, that provides meaning for the results. And I encourage you to see subsequent webinars for more discussion and articulation of that. And finally, and hopefully I've emphasized sufficiently in this brief time with you, as with all outcomes, PROs are best understood in the context of overall treatment efficacy, treatment safety, the preferences of patients that do vary, the economics of the treatment, and the impact of treating or not treating on society overall. So thank you, and good luck to you.